The next topic is enzymes for manipulating DNA or molecular tools used in recombinant DNA technology. First is the restriction endonuclease. This we have already discussed. Then ligases, DNA polymerases, alkaline phosphatases, nucleases, and topoisomerases. Now we'll discuss one by one. Ligases. Ligation reaction means joining two molecules together by forming phosphodiester bond between two. DNA ligases catalyze formation of phosphodiester bond between 5' phosphate of one strand and the 3' OH of other to permit joining of the two DNA molecules. The most widely used DNA ligase is derived from T4 bacteriophage named T4 DNA ligase. This T4 DNA ligase requires ATP for its action, requires ATP as a cofactor, and it also requires double-stranded DNA for joining. T4 RNA ligase can use single-stranded RNA or single-stranded DNA substrates. Now here the ligation reaction is shown. This is one segment of DNA and this is another segment of DNA, double-stranded DNA. This has free 3' OH and here is free 5' phosphate. So the phosphodiester bond forms between OH, 3' OH and 5' phosphate, and with the help of ATP and Mg2 plus T4 DNA ligases join these two segments of DNA. Ligases are used for lagging strand DNA synthesis after removal of primers. When the gap is filled, the gaps are joined, the two DNA segments are joined by ligation reaction or ligases enzymes. It is required for genetic recombination, cutting and exchanging the segments of DNA, needed for DNA repair, uses energy from ATP, and works on blunt or sticky ends. T4 DNA ligase requires ATP, while E. coli DNA ligase require NAD+. So in each case, the cofactor is split and forms enzyme AMP complex. Cofactor forms enzyme AMP complex. The complex then binds to the nick. Nick is the single-stranded break or simply the break present in DNA molecule, which must expose 5' phosphate and 3' OH and makes covalent phosphodiester bond between two. The optimum temperature required for ligation reaction is 37 degrees, but at this temperature the hydrogen bond join between the sticky ends is unstable. So mostly ligation is carried out at lower temperatures, 4 to 15 degrees Celsius overnight. Thus is ligation reaction. See, here is the single stranded break, because here you can see free 3' OH and phosphate. Here is the gap between these two segments, which is known as nick. Nick means gap present between two DNA segments. The enzyme where it is T4 DNA ligases requires ATP, E. coli DNA ligases require NAD+. Okay, so both NAD+ and ATP, they are converted, they form enzyme AMP complex by releasing their components. They form enzyme AMP complex. This enzyme AMP complex then joins the nick or bind to the nick where 3' OH and 5' phosphate groups are exposed. Then AMP is released out and enzyme join two DNA strands together by forming phosphodiester bond. Same reaction is given here. This is in repair reaction. Where the nick is present or gap is present, enzyme joins 5' phosphate and 3' OH. Next enzyme is alkaline phosphatases. The name itself suggests phosphatases, that is, it removes phosphate group. Removes phosphate group. Phosphatases remove phosphate group, and kinases add phosphate group. Both are different enzymes. Their actions are completely opposite. Alkaline phosphatases remove 5' phosphate group. Now where is the phosphate group present? On the 5' side of the DNA molecule, or wherever the break is there, to the 5' side phosphate group is present at the end of the DNA molecule. This alkaline phosphatases remove 5' phosphate and prevent formation of new phosphodiester bond by DNA ligases. This is useful for treating restricted vector DNA sequences prior to ligation and thus it prevents religation of the vector in absence of insert DNA. See, if you cut the vector, there must be a diagram. See this one. If you cut a plasmid vector with the restriction enzyme, what happens is, I'll show you. Say suppose this is the plasmid vector. Consider this as double-stranded circular DNA. If you cut it with the restriction enzyme, this will generate two ends. One is 5' phosphate and one is here is OH. So before joining this targeted DNA or say foreign DNA to this vector here in between, these two can rejoin and can form plasmid like this again, because these already have 5' phosphate and OH. Ligases can join this one without joining this foreign DNA here in between. So to prevent this, these phosphate groups are removed by treating them with alkaline phosphatases. So all the ends, they will have OH. Since phosphate group is removed, what will remain is OH. So this OH cannot rejoin together to form a circular DNA. Okay. Then this vector DNA, when mixed with the target or foreign DNA or a gene of interest, these can join like this. Now what will happen is, see, these all have, for a plasmid DNA, vector DNA have OH on all the sides. Okay, see, this is double-stranded DNA, and this target DNA, this has 5' phosphate and 3' OH. Here is 5' phosphate and 3' OH. So this one and this one will join with the OH, but these OH won't be joined. Sorry. So see here this one can be joined easily with the help of ligases, and here this end is joined easily. Here this remains as it is and here this remains as it is, because both the sides they have OH groups. So these ends are again treated with the 5' phosphate. These ends are again treated with the kinase enzyme. Kinases will add phosphate group at these ends, and then this 5' phosphate and 3' OH can rejoin. See here, this plasmid treated with the restriction enzyme and alkaline phosphatases, so both the ends will have hydroxyl groups. This is the foreign DNA. When this foreign DNA is mixed, one ends are joined easily and other remain as it is, since both the ends have OH. So these are treated with the kinases. Again, phosphate groups are attached so that they can be ligated easily. The next enzyme is polymerase. As we have studied in replication, transcription, DNA and RNA polymerases, they polymerize nucleic acid. DNA polymerase can polymerize or can join DNA molecule, can synthesize new DNA molecule. RNA polymerases can synthesize RNA molecule from the direction 5' to 3'. Okay, DNA polymerase require primer and RNA polymerase does not require any primer for its action. Reverse transcriptase is a type of RNA dependent DNA polymerase which synthesize double stranded DNA using RNA molecule. Okay, double stranded DNA using RNA molecule. Then the next type of DNA polymerase is TDT. This is used in most of the recombinant DNA experiments. Terminal deoxyribonucleotide transferase. Terminal deoxynucleotide transferase, TDT. This enzyme, irrespective of template, add single nucleotide, many single nucleotides at one end, either thymine or adenine or guanine or cytosine. Okay, this is template independent enzyme, do not require any template. This adds single nucleotide many times independently. Okay, DNA polymerase 1. This is the special enzyme used in gap filling or nick translation reaction. This enzyme, DNA polymerase 1, has 5' to 3' exonuclease activity. 3' to 5' exonuclease activity is present in all DNA polymerases, but only DNA polymerase 1 have 5' to 3' exonuclease activity. The DNA poly 1 can be fragmented in two parts. One is larger segment and one is smaller segment. Larger segment is known as linopolymerase. Okay, discovered by scientist Lino. This larger segment lacks 5' to 3' exonuclease activity. Therefore, it has only polymerization activity. It can only synthesize nucleotides and proofreads it, because 3' to 5' activity is for all the DNA polymerases. This is used, Lino fragment is used for labeling of DNA termini by filling in cohesive ends generated by restriction enzymes, used for DNA sequencing and for generation of blunt ended DNA molecules or linkers. E. coli DNA pol 1 Lino fragment. It has 5' to 3' DNA polymerization activity. So, see, this is the DNA strand, and this strand is extended with the help of DNA pol 1 lino fragment. DNA pol 1 also has 3' to 5' exonuclease activity. This can remove bases from the 3' sides as well as from the 5' side. It can remove exonuclease 3' to 5' exonuclease, means from this end to this end it can remove nucleotides. And if, see here, if there is a gap, or simply it can add labeled nucleotides with its polymerization activity. See here, this is used for labeling the ends of the DNA molecule or preparation of probes. T7 DNA polymerase. Next is T7 DNA polymerase. It is highly processive with highly active 3' to 5' exonuclease activity. This is used for labeling of DNA termini. Modified T7 polymerase is used for sequencing reaction because it lacks both 3' to 5' and 5' to 3' exonuclease activity. They are also termed as sequenase because it is used in sequencing reactions. These are used for PCR, Taq polymerase, PFU polymerase and Vent polymerase. Taq polymerase is extracted from Thermus aquaticus. PFU is extracted from Pyrococcus furiosus. This have lowest error rate, and Vent polymerase is extracted from Thermococcus littoralis, also known as TLI polymerase. Half-life of this Vent polymerase is higher than Taq polymerase. Nucleases, they are categorized into DNases and RNases, also exonuclease and endonuclease. Exonuclease, which cut the DNA or RNA from ends. Endonucleases cut the nucleic acid in between. So these are all basic things. You can just simply read. DNase 1 is used mostly in recombinant DNA technology experiments. This works in presence of manganese and magnesium ions, and this cleaves single-stranded as well as double-stranded DNA. So this DNase 1 is used for eliminating DNA from preparation of RNA, analyzing DNA protein interactions and nicking DNA prior to labeling by nick translation. RNase A, it is a type of ribonuclease which cuts single-stranded RNA at 3' end of pyrimidine. It degrades RNA into 3' phosphorylated mononucleotides and oligonucleotides. These are used for reducing RNA contamination during plasmid extraction, mapping mutations in DNA. RNases cleave RNA in DNA-RNA hybrids. This we have already discussed, exo and endonucleases. Topoisomerases is used for reducing the strain onto the DNA. kinases, polynucleotide kinase is used for phosphorylation. See here, these are the DNA ends treated with the phosphatases. Now the 5' ends are again converted to 3' ends with the help of polynucleotide kinase enzyme. See here.